This video will cover subtopic D.2 Digestion from the physiology option. Here are the list of understandings, application, and skills that will be covered in this video. So we'll start with control of the digestive system by the nervous system and hormonal control of the digestive system. So the digestive system is basically under control of the autonomic nervous system. And the autonomic nervous system is what controls body processes that are not under our conscious control. So we don't have conscious control over them, they just kind of happen. Now, the digestive system can also be controlled by hormones released by glands in the body, such as salivary glands. The autonomic nervous system includes the sympathetic nervous system and the parasympathetic nervous system. These two branches of the autonomic nervous system are always in competition with each other, and they're kind of antagonistic in a way. They're always competing for control. So the sympathetic nervous system is responsible for that flight or fight response of the body. So when we get scared, the sympathetic nervous system will increase our heart rate and will provide energy to our muscles in preparation for action. So we'll either stand our ground and fight, or we'll turn and run. In order to provide more energy to the heart, lungs, and muscles, the sympathetic nervous system will inhibit, it will basically slow or stop digestion. So it will look to slow down digestion, and it does this by directing blood flow away from the digestive tract and redirecting that blood now to our lungs and our muscles, where it will be needed for that quick action. The parasympathetic nervous system is responsible for stimulating digestion by increasing blood flow to the digestive tract. The parasympathetic nervous system will stimulate salivary glands to secrete and will also increase peristalsis. This will increase digestion of food and absorption of nutrients. Remember, peristalsis is those rhythmic contractions of the intestinal tract that will move a food bolus through. So this diagram just shows the contrasting functions of the parasympathetic nervous system, the rest and digest, and the sympathetic nervous system, that fight or flight response. So, in relation to digestion, that's parasympathetic nervous system will stimulate stomach activity. It will also inhibit the release of glucose, stimulate the gallbladder to produce or to release bile, and will also stimulate activity of the intestines. Whereas the sympathetic nervous system, when looking at the digestive system, will inhibit that stomach activity, inhibit activity of the intestines, and mainly doing this through redirecting blood flow away from the digestive tract, whereas the parasympathetic nervous system would direct blood flow to the intestinal tract. So in the digestive system, there are glands referred to as exocrine glands, and exocrine glands are going to be glands that secrete their products into ducts. So this is different from endocrine glands, which secrete directly into the bloodstream. So remember, like the corpus luteum, for example, would be considered like an endocrine gland, as it's secreting progesterone directly into the bloodstream. However, exocrine glands, like salivary glands, for example, would secrete their products into ducts that basically get released outside of the body. So in the digestive system, these glands can include salivary glands, the pancreas, and those that line the stomach and intestines. With exocrine glands, the products that they release, the composition of each of those secretions will be different. And more information on this can be found on page 672 in your textbook. The structure of an exocrine gland has numerous endoplasmic reticulum for enzyme synthesis and mitochondria to provide ATP energy. So, these exocrine glands are producing a lot of proteins, a lot of enzymes, for example, that get released from the cell. So all of that requires a high number of ATP. So there's lots of mitochondria to provide that energy, lots of endoplasmic reticulum and even Golgi as well to allow all that protein to be synthesized. And the diagram here shows kind of a basic structure of an exocrine gland. So this circular shape here is the exocrine gland. These little pizza slices would be an exocrine cell. So the purple circle is going to be the nucleus. And then these openings here between these cells, these are going to be the ducts. So these exocrine cells will release their products into the duct, and these products will then travel through the duct. So if this is a salivary gland, for example, this duct would then empty out into the mouth, for example, and this is where saliva comes from. So exocrine cells will also have a large number of secretory vesicles, since they are releasing products outside of the cell into those ducts. So looking at the epithelium now of the villi and the specific structure of the epithelium on the villi, that epithelium contains what are called tight junctions made up of tight junction proteins. So the tight junction of the villi will ensure that most material passes into the blood vessels of the villi and does not pass between the cells. So in our picture here of the small intestine, so if this is the villi here, this area here is magnified to show these epithelial cells. These epithelial cells have what are called tight junctions. They're basically like proteins that connect the two cell membranes together or hold the cell membranes close together. And so contents in the lumen cannot pass between the cells, but any contents traveling from the lumen into the blood vessels below have to travel through the epithelial cell. And this is how absorption of nutrients will happen. So the tight junctions ensure that nothing passes between the cells, but instead has to pass through the epithelial cells. Now the intestinal lumen also has extensions, or those epithelial cells will have extensions on them called microvilli, which are on the intestinal side and are termed the brush border. So these epithelial cells have these little folds on their membrane which faces towards the lumen, and these folds will help increase the surface area through which absorption can occur. But because they kind of look like a brush when viewed under a microscope, they gets termed as the brush border. High amounts of ATP are required to drive active transport, which require large numbers of mitochondria found in these epithelial cells. And then the various sides of the epithelial cells do get special terms. So this side here, facing the lumen, so like the side with the microvilli, is referred to as the apical surface. Here it is in the diagram, the apical surface. And then the surface facing the blood vessel, so if this is our epithelial cell again, we have the lumen here, and then just below this is going to be the blood vessels, just beneath the villi. This surface that is facing the blood vessels is referred to as the basal surface. These surfaces have different types of proteins for material transport. So there are different transporter proteins located on the apical surface and different proteins located, different transport proteins located on the basal surface of these cells, as what they are bringing in or what they are allowing in or what they are transporting in may be slightly different from what they are transporting out. But you'll find, or the method in which items are transported in are different from how they are transported out. But either way, those proteins are going to be different. So those transporter proteins are different on the different surfaces. One of the skills that you will have to be able to do is to be able to identify exocrine glands that secrete digestive juices from electron micrographs, and as well identify villus epithelium cells from electron micrographs. And more information can be found about this on page 674 of the textbook. So looking at an electron micrograph now of an exocrine cell, and this is a pancreas, what's called an acinar cell, but it's an exocrine cell, you can see that there are a high number of rough endoplasmic reticulum in the cell. There are a few mitochondria that can be identified, and then there are these vesicles. So key features to look for when trying to identify an exocrine cell. When identifying a villus epithelium, generally speaking, in those electron micrograph pictures or in a light microscope picture, what you're looking for is basically the structure, keeping in mind the structure of that villi. So that villi is going to have a single layer of cells, and this single layer of cells are going to be the epithelium cells. And the individual epithelium cell will just have one nuclei to it. And then depending on the type of staining, you may see multiple mitochondria in there. Some cells will have these clear structures within them, these clear vesicles, and these cells are specialized cells referred to as goblet cells, which secrete mucus on the outside of the villus. So within the stomach, we do have to consider the conditions found within the stomach, which are going to be acidic. So the main component of gastric juice is going to be hydrochloric acid, which is produced by a type of cell called a parietal cell in the gastric glands of the stomach. So they are glands within the stomach that produce hydrochloric acid, and these are going to be exocrine cells, essentially, because they are secreting hydrochloric acid into the lumen of the stomach. These types of cells found in the glands are referred to as parietal cells. Another function of the hydrochloric acid is that it will denature proteins, so it changes the pH conditions, those proteins unfold, exposing the single polypeptide chains. In the stomach, we find an enzyme called pepsin, which is produced in the stomach and can actually break down or cleave the exposed peptide bonds within the polypeptide chain. So pepsin is released by a type of cell referred to as chief cells in an active form referred to as pepsinogen. The acidic conditions within the stomach will convert this inactive pepsinogen into the active form of pepsin, where that enzyme can then do its job. Now, within the stomach, there can be certain conditions that arise. One of them is due to the development referred to as ulcers. So, ulcers occur when the stomach acid make contact with the stomach cells. So there is often a mucus layer that separate those stomach cells from the acid produced in the stomach, and erosion of that mucus layer can lead to the development of an ulcer. So development of ulcers has been linked to a particular type of bacteria called Helicobacter pylori. And this was a discovery that was made, I guess, a few decades now ago. where this bacteria was actually found to be a contributing factor in the development of ulcers. So people that had stomach ulcers, scientists were finding oftentimes that this bacteria was present. But there was no kind of definitive proof or definitive way of showing that the bacteria caused the ulcer. You know, it was a big question of whether it was just coincidence that the bacteria were present in people with ulcers. The scientist who pieced this all together actually took a sample of Helicobacter pylori, ingested it, went on to develop stomach ulcers as a result, treated himself with antibiotics, cleared out the Helicobacter pylori infection, and actually cured himself of those ulcers. So he showed that definitive link that Helicobacter pylori cause stomach ulcers. And the presence of stomach ulcers greatly increases the risk of developing stomach cancer. So oftentimes there can be overproduction of the gastric juice, that hydrochloric acid, which can cause damage to the esophagus or, as you saw in the previous slide, the development of ulcers in the stomach. So sometimes medication is required to inhibit the amount of acid that is produced. So these medications are referred to as a proton pump inhibitor. Remember, it's hydrochloric acid in the form of protons that gets pumped into the lumen of the stomach, and these medications will prevent these protons from being pumped into the lumen. So a reduction in the amount of stomach acid produced can be controlled by medication. Remember that stomach acid is corrosive and can destroy the lining of stomach, causing an ulcer. And proton pump inhibitors basically work by irreversibly blocking the hydrogen ion, potassium ion, ATPase enzyme of the gastric parietal cells. So this particular pump, and remember it's a pump because it will use ATP, this pump will use one ATP to exchange two protons from the cytoplasm of the parietal cell with two protons from the cytoplasm with two potassium ions from the lumen. So basically what this cell is doing, if that's our cell, here's our nucleus, it's taking hydrogen ions that's found inside of the cell and pumping them into the lumen in exchange for potassium ions found in the stomach, taking those into the cytoplasm. And because there's going to be a high gradient of protons in the lumen of the stomach, a pump using ATP is required to move these hydrogen ions or these protons from an area of low concentration to an area of high concentration. So proton pump inhibitors will prevent this from happening. So the amount of hydrogen ions or protons pumped into the lumen gets reduced. And there are a variety of drugs that can do this. Zantac was one famous kind of over-the-counter brand, which has recently been pulled due to other issues that it can cause. So in studying the stomach, one name stands out in particular, and is that of William Beaumont. He is considered the father of gastric physiology. So William Beaumont was a physician during one of these wars, I don't remember which one, but basically the story goes is that there was a soldier who suffered a wound, and the image is kind of shown here, the recreation of it, suffered a wound in the side of his abdomen. It wasn't fatal, but William Beaumont basically used this individual. He didn't heal the wound, but kind of kept that exposed wound. And that exposed wound led into the stomach. So we used that exposed stomach to study the process of digestion. So that wounded soldier who had that gaping hole in his side, he used that soldier to study that process of digestion. So basically what he did is that at different points or whatever, he would take a string, and that string was attached to a piece of meat. And through that hole, he would place that piece of meat into the stomach of that individual, And at various time points, he would remove the piece of meat through that string, through that hole, and would study the composition of that meat. And through this process, he showed that digestion in the stomach involved chemical action, overturning the idea that it was only physical. The prominent idea was that the stomach just churned and mushed that food around, but he showed that the gastric juice produced in the stomach was critical in promoting digestion of that food. So with egestion now, and egestion, so if things come out, or things go in, then things have to come out. So with egestion now, deals mostly with fiber in regards to egestion. But so fiber, which is also known as cellulose and lignin. Remember, cellulose is the part of the plant that cannot be broken down or absorbed. That cellulose is what goes to make up the plant cell wall. Is basically, that fiber is going to be excreted along with bilirubin, which is a major component of feces. It's going to be excreted along with that fiber and water in the form of feces. So what fiber does now is that it will increase the bulk material passing through the intestine and will help prevent constipation as it draws water into the intestine. So fiber will work to increase the transit time down the digestive tract. So meaning like it'll increase the time it takes from the time you eat something to the time you eliminate it. That transit time will be increased, so things will pass through quicker. The presence of fiber in the stomach may also help to increase that feeling of fullness and reduce the need to eat more. And food of plant origin will contain dietary fiber. Looking at another disease now of the intestinal tract, which is cholera. Cholera is an infection of the small intestine by some strains of bacteria referred to as Vibrio cholerae. The classic symptom of cholera is basically large amounts of watery diarrhea that lasts a few days. The diarrhea can be so severe that it leads within hours to severe dehydration and electrolyte balance. The severe dehydration and electrolyte imbalance can actually lead to death. People lose so much water so quickly that they just basically kind of go into shock and then die. So the way that cholera works is that the toxin, it produces a toxin that binds to receptors on the intestinal cells, triggering a cascade response that ultimately leads to the efflux, the outward movement of chloride ions and carbonate ions, or bicarbonate ions, into the intestines. Water will follow by osmosis, so you have a high ion concentration in the lumen versus inside the cell. Water will move into the lumen now to kind of balance out the concentration, which leads to this watery diarrhea. The bacteria is basically looking to spread, so once it gets into its host, the idea is it wants to reproduce and then spread to other people. So to do this, it triggers this diarrhea response. So it wants this diarrhea in order to be spread. So as that water flows into the intestinal tract now, that water is drawn from the blood into the cells to replace the fluid lost. And if this happens like very frequently, ultimately, the water content within the blood just drops too low. So this diagram just goes through how cholera affects the body. So how it spreads is that people will ingest water or food contaminated with cholera bacteria. And in epidemic, when there are epidemics of cholera, the feces of diseased person is a source of contamination. So basically, the only treatment that there is salt solution, intravenous fluids, and oftentimes antibiotics can help. But you want to try and provide the individual being affected with electrolyte solutions. So like Gatorade is good. Not ideal, but works in a pinch. In unprepared communities, the death rates can be as high as 50%, which is like really high. And so cholera can be problematic in poor areas where water is untreated, or developing areas, where they don't have reliably clean water sources. Also, cholera can be a problem after, let's say, a hurricane, or there's like a severe catastrophe in an area, and water treatment is not available, or there's large flooding in an area. Then there's always a high risk of cholera spreading amongst that population. So that is the end of this video for D.2 digestion.